Yeah, intelligent iNKT cells. Actually, this intelligence really relates to cells as opposed to antibodies, because antibodies are one thing, they're a molecule, they're not active beyond what they intrinsically recognize and activate in the immune system. Cells actually have, by their very nature, and not just iNKT cells, but all immune cells, a sense of awareness of where they are and the context through which they get activated, which actually is a very good basis for an indication like cancer, because it's a multifaceted disease that you really need to attack on multiple fronts to make an impact. So cell therapy is really about finding those cells that you can then use and retarget and repurpose for, in our case, cancer treatments or infectious disease treatment. And the sort of the properties you're looking for is a way to get to where the disease is, which is tumors, for instance, tumor homing, and also respond to certain signals in the tumor environment, the cancer cells that are there, but also the stromal and the myeloid suppressive cells, for instance, and ideally attack the cancer on multiple fronts. And that's where sort of this idea of intelligence in a cellular form comes from, because what you're trying to build is a platform that is actually able to get into the tumor and respond appropriately and really change the tumor into an environment that can lead to its eradication and long-term cure. Not alone, obviously, because you need the endogenous immune system to help you there. So you need to be able to attract other cells into the tumor to help. And you need to sort of reset, ideally, the endogenous immune system to recognize this cancer going forward so that you don't get it back, actually. And that's quite a task, but that's actually what Immuno-Oncology has been all about in the last sort of decade or so, since ipilimumab actually got onto the market. I have a history in antibodies, and I know that antibodies can have a fantastic effect on the systemic immune system, but sometimes the endogenous immune system is just not capable enough at that particular moment in time to really fight cancer. So you need to add something, which is cell therapy. And you need to do this in a way that you can set it up to be a success in conjunction with the rest of the immune system. So that's really why we think these iNKT cells are a fantastic basis for it, because they have properties that are bridging the innate immune system. So they have a number of NK cell properties, but they're actually T cells. So they have a T cell receptor. And in this case, a very particular T cell receptor that is invariant, which basically means your T cell receptor is the same as my T cell receptor for iNKT cells, which means you can apply it allogeneically. You don't have to edit out the T cell receptor to make it not cause graft versus host. Invariant NKT cells do not cause graft versus host because they recognize an invariant HLA molecule. So that already sets them up for an allogeneic platform approach. These cells do not need any editing to be accepted without causing graft versus host. And they also are tissue resident, so they home to tissues naturally, which in our mind makes them a very good basis for developing therapies for solid tumor in this case. Of course, these cells have a lot of native properties of their own. They have NK properties. They have this particular T cell receptor that homes to CD1D, which is a molecule that is expressed in a variety of tumors, both on the cancer cells as well as on the myeloid compartment. So we really need to understand what these cells do by themselves and then start enhancing their efficacy. So that's the basis of our first clinical trials. And to start a trial, you need to be able to manufacture these cells. And the sort of special property of these cells are they're not abundant in the blood, but they're there. You can isolate from a leukopak up to sort of 5-10 million of these invariant NKT cells. And they expand massively. They are proliferating beyond what we have seen from normal T cells or other cells. And you can actually get from a couple of million cells to about 20, 40, 50 billion cells in about 30 days. And they do not exhaust during that process. And that's a very, very important basis for any cell therapy. If you can't manufacture, you can't treat. So in our case, being able to manufacture and having that process down to a robust, repeatable process is a very good basis for that. And we've used that to now go into the first clinical indications to really start seeing how these cells impact these diseases. And in our case, we've started a trial in COVID, because these cells, being immune regulators, also have a way to orchestrate an immune response and add their efficacy to viral infections, both by attacking viral infected cells, but also by dampening the runaway immune responses that you've seen happening in late-stage COVID patients. So we're currently have been treating 5 patients initially, and there are going to be a couple more, quite a few more. And then we will see if the effect that we expected these cells will have in both controlling the inflammation and sort of keeping the runaway immune system in check really has a benefit for these patients, as we would expect them to have. In parallel, we've started up a trial in multiple myeloma, and that it really is to see the direct impact of these cells on a disease that actually expresses CD1D and also is present in the bone marrow. What we do know of these invariant iNKT cells is that they very effectively home to the bone marrow, among other tissues. So looking at a bone marrow resident disease as a first indication to check their impact, multiple myeloma comes to the top of the list on that one. So those are 2 trials that we are starting. We do want to get into solid tumors as soon as possible, both because they are a large unmet medical need. Still, there's a lot of cell therapies for leukemia, lymphoma, but there's not yet many effective or any effective therapies for solid tumors. These cells being tissue resident and homing to tissues actually would form a very good basis to develop therapies for solid tumors. And that also actually brings us to these combinations. Agenus is a fully owned subsidiary currently of Agenus, and Agenus has an antibody portfolio of checkpoint modulating antibodies. The furthest advanced are the anti-PD-1 and anti-CTLA-4 antibodies, which are going to go into a BLA soon. And it's an obvious combination with cell therapy. You do expect just to retrace, so this iNKT cell therapy is an allogeneic therapy, which will have an impact in the initial phases, but eventually over time, these cells will disappear from the patient. And when they disappear, after having made their impact on the tumor, you do need the endogenous immune system to take over and stay vigilant. And that is actually where checkpoints come in. They've proven to be able to reactivate elements of the endogenous immune system in cancer patients. And also, in the case of CTLA-4, they can lead to priming of new cells and elimination of T regulatory cells. So together with cell therapy, allogeneic cell therapy, which will disappear over time, these checkpoints will then re-educate and reinvigorate parts of the endogenous immune system to enable the cancer to stay under control. So we feel that it's important to investigate the contribution of checkpoints on top of cell therapy. And that's why we're aiming to get into solid tumors as soon as we can, which we aim to be this year in a number of indications that, and also then embark on combination trials soon thereafter at the same time. So that's roughly where we are with the programs.